Hello, thank you for having me. I'm James Morton, and I'm a research scientist at the Simons Foundation, and today I'm excited to share my work in joint collaboration with Justin, Gleb, Hari, and Rich, where we show how variational autoencoders can enable scalable inference to infer massive microbial co-occurrence networks spanning across tens of thousands of biological samples. But before I begin, let me give some motivation why we care about microbial ecosystems. It turns out that microbial ecosystems have a very real impact on our daily lives. Our gut microbiome serves as a bioreactor, providing the rest of our body the essential nutrients in order to function. When its function breaks down, it can lead to a multitude of health issues ranging from gastrointestinal issues to neurological disorders. Furthermore, environmental microbes drive global processes such as nitrogen fixation and has a real impact on global warming. So what exactly are these microbial ecosystems? When I talk about microbial ecosystems, you may draw connections to macroscopic ecosystems where plants, fungi, and animals interact with each other through competition or predation, forming complex food webs. But you might not draw the parallel to a well-oiled car motor. But there is one thing that these two systems have in common, namely, they are both dynamical systems. Furthermore, if the fundamental theories of ecology apply to microbial ecosystems, then given enough information, we can manipulate microbial ecosystems and predict outcomes in the same way that we drive cars and predict its current trajectory. But if we want to put these ecological theories to the test, we need to estimate the underlying species-species interactions of these microbial ecosystems. When we're trying to obtain insights from microbial ecosystems, we often process samples using DNA sequencing to count the number of microbial species within a sample. As an example, the resulting data is given like below, where the samples are rows, the microbial species are columns, and the values within the table represents the number of times a microbial species has been observed. However, the data that we collect are proportions which should live in a sample space called the simplex. Most of the existing statistical methods are not designed for these types of data, and as a result, many of the statistical tools that we apply to microbiome data are inappropriate. Without the ability to quantify the total abundance or the scale of each sample, the resulting data is said to be compositional. And discussions surrounding compositional data have proliferated across many other fields, including geology, political science, as well as macroecology, RNA-seq, and metabolomics. To see some of the challenges dealing with compositional data, consider the following. Suppose that we have a microbial ecosystem of 3 species, where the blue and orange species are both increasing and the green species is flatlined. From this, we can conclude that there is a positive correlation between the orange and the blue species. However, in most microbiome studies, we do not have access to the total abundances and are stuck analyzing the proportions, which yields a completely different story. Now we see that the blue and the green species appears to be decreasing and that there is a negative correlation between the orange and the blue species. The takeaway from this is that we cannot infer microbial correlations from proportions alone. We need additional information, such as total abundance or cell concentrations, in order to estimate these correlations. Unfortunately, due to technical constraints, the vast majority of studies do not have such information available. So here's the question. Without this information, what can we infer? In this talk, I would like to address 3 questions, namely, how can we infer microbial co-occurrence patterns from biological specimens? And how can we use this information to construct meaningful representations of ecosystems? And finally, can we make inference scalable across many biological studies? To address these questions, I'll first introduce the multinomial logistic normal distribution and the challenges of estimating it. And then I'll make the case that carefully crafted variational autoencoders, or VAEs, can quickly estimate these distributions on high-dimensional data. Finally, I'll show how biologically relevant information can be extracted from these neural networks. So what is the multinomial logistic normal distribution? Suppose that we have a biological specimen that we've sequenced, and from this we obtain d-dimensional counts, where each dimension represents a microbe. That sample is said to be distributed from a multinomial logistic normal if it can be explained from the following generative model. Namely, we model the sequencing counts from a multinomial distribution, and that the parameters of this multinomial can be generated from a normal distribution after applying softmax. This softmaxed normal distribution is also known as the logistic normal distribution. The multinomial logistic normal has multiple nice properties. First, it is scale equivariant, which means that we don't need cell concentrations or totals in order to estimate the parameters of this distribution. It'll work with any dataset that is consistent of counts. Second, because of the multinomial aspect, zeros and low count uncertainty can all be modeled with this distribution. Finally, this distribution can handle a more complex correlation structure. In contrast to the Dirichlet distribution, we can handle correlations beyond just sampling variability. One of the subtle issues of utilizing this distribution actually boils down to the definition of softmax. You see, softmax has an identifiability issue, which you may know as the softmax trick. This is actually problematic, since we won't be able to identify the underlying covariance matrix of microbial correlations that we're interested in. This identifiability issue can be easily fixed by constructing a linear mapped psi, which will consequently restrict the covariance matrix to a d-1 dimensional space. If psi is orthonormal, then this new transformation is known as the inverse ILR transform. This will later prove useful when extracting principal components. It turns out that estimating the multinomial logistic normal distribution in higher dimensions is incredibly challenging for two reasons. First, when we consider just the logistic normal distribution, that distribution does not have any analytically tractable expectations. As a result, expectation maximization is a dead end. Second, the multinomial logistic normal distribution itself does not have an analytical probability density function. As a result, we can't even do standard maximum likelihood estimation. To showcase how difficult this problem is, here's a timeline of all the methodologies that have attempted to estimate the multinomial logistic normal distribution in some shape or form. The first attempt started around 2001, where Bill Heimer developed a Gibbs sampler to estimate this distribution across 30 samples. Around 2013, we see there is renewed interest, but most methods were not able to scale beyond 100 dimensions. In 2019, there are two methods that use a Laplace approximation and a customized Hamiltonian Monte Carlo algorithm to bring this up to roughly 1,000 dimensions. However, it is important to note that the average microbiome dataset is consistent of 1,000 dimensions, and these algorithms are just able to handle the average dataset. In the rest of this talk, I'm going to make the case that we can now scale inference several orders of magnitude faster using VAEs. This will allow us to scale inference to handle not just one dataset, but handle dozens to hundreds of biological studies. Our VAE architecture is split up into two parts. The encoder architecture to perform the dimensionality reduction of the biological samples, and the decoder architecture to reconstruct the data given the latent representations. To describe why these VAEs can't estimate the multinomial logistic normal, I'm first going to make the case that our decoder architecture directly corresponds to multinomial probabilistic PCA. When I refer to multinomial probabilistic PCA, I'm referring to the following generative model, where the data is generated from PPCA and is passed into the multinomial parameters in order to generate the sequencing counts. For those of you not familiar with probabilistic PCA, this is a model that provides a probabilistic interpretation to principal components analysis. This allows us to quantify measurement uncertainty after performing dimensionality reduction. In addition, probabilistic PCA allows us to approximate the covariance matrix as a low-rank decomposition. This formulation of multinomial probabilistic PCA itself is novel and advantageous because it allows us to apply principal components analysis directly to count data. Ultimately, this allows us to avoid cubic runtime matrix inversion operations, which is commonly used in previous multinomial logistic normal estimators. Despite this novel model formulation, we are still stuck with intractable integrals when we try to perform an estimation, and this is where VAEs can become handy. The VAEs offer two major advantages. First, rather than memorizing the latent representation for each sample, as done in probabilistic PCA, we can use a neural network to directly estimate the latent representation. Specifically, we can use neural nets to estimate a distribution of plausible latent representations for each sample using the biological data at hand. This is nice since we do not need to retrain these models every time we see new biological samples. And this is also great for out-of-distribution generalizability, since these neural networks can interpolate across unobserved samples without having to update the rest of the model. The second major advantage is the ability to draw differentiable Monte Carlo samples through reparameterization gradients. When we estimate the parameters of these VAEs, we try to maximize the evidence lower bound, or the elbow. And we can break this elbow into two parts. The second part is simple, since it is a closed-form solution. But the first part of this elbow would be difficult to estimate due to the expectation. We can estimate this expectation using Monte Carlo sampling, which is relatively straightforward to compute. But the major breakthrough comes from Kingma and Welling when they realized that it is possible to differentiate through these Monte Carlo samples and enabling backpropagation to the rest of the neural network parameters. This is the reparameterization trick. So while we cannot evaluate these expectations, we can approximate them through these reparameterization gradients, allowing us to use stochastic gradient descent to obtain an optimal set of weights. And the best part about this is that the objective function is actually well behaved, and if we use a single layer encoder, the optimization landscape is biconvex. With simulations, we have observed that the VAEs can exactly recover the ground truth principal components on dense data with a Pearson correlation of 0.96. When we apply these VAEs to a sparse data set with 40% zeros, we see that there is some performance degradation where the Pearson correlation drops to about 0.7. But more importantly, we see that the inverse Euler transform was critical for accurate covariance estimation, and that there is a large gap between the inverse Euler transform and other transforms, including the standard softmax transform and the inverse ALR transform. There are some subtle geometric considerations that explain this performance difference, and I won't discuss it in this presentation, but feel free to follow up with questions afterwards. In practice, we see that these VAEs are competitive with existing beta diversity metrics. In the microbiome literature, beta diversity refers to the idea of measuring biological samples through distance metrics. And with these distance metrics, we can evaluate the performance using k-nearest neighbor classification accuracy. In our case, we looked at mouse data sets to predict experimental outcomes. And as we can see here, that the performance of the VAEs is comparable to some of the best performing distance metrics. And the performance difference is particularly noted on the longitudinal data sets, where the VAEs outperform other methods by upwards of 10% classification accuracy. This isn't unexpected, since compositional methods are known to perform better on longitudinal data sets. On the other hand, we see LDVAE, another linear VAE developed for single-cell RNA-seq data, drastically underperforms all of the methods here. And this highlights the importance of considering the underlying geometric constraints of the data when designing these models. Up to this point, I haven't said anything about microbial interactions. While we can because of the way we designed the VAE, we can directly extract co-occurrence information from the decoders. And it turns out that the transformed decoder distances are directly proportional to the variance of the log ratio of their counts. Specifically, if we focus on 2 microbes and compute log ratio of their counts for each sample, the variance of the log ratios is given here. And this quantity will be proportional to the corresponding decoder distance. We can postulate that this is due to the connections between the VAEs and the probabilistic PCA. This variance log ratio can be useful for inferring correlations. If the quantity is small, then that suggests that those 2 microbes are commonly found at the same ratio across samples. This quantity is known to be scale invariant, where you don't need to measure the cell concentrations at all for this quantity. However, there is one caveat to this metric. Namely, it cannot handle 0 counts, since the log of 0 is not defined. So to see if the decoder distances really do correspond to the variance log ratio in reality, we can focus on only the top most abundant microbes. Indeed, we see that there is a strong correlation between the decoder distances and the variance log ratio. This gave us more faith in interpreting the VAE decoder weights, and we went ahead and investigated all of the top co-occurring microbes detected by the VAE. Interestingly, we see that if 2 microbes are tightly co-occurring with each other, they have very little chance of being phylogenetically similar, or even having similar metabolism. Biologically, this makes sense to us, because phylogenetically similar microbes tend to compete with each other. And since they have identical enzymes, they'll consume the exact same resources. What makes this really exciting is that this observation was independently validated in a huge microfluidics experiment measuring upwards of 180,000 microbial interactions, giving additional ammunition that our VAEs do work. To summarize our findings, we have provided a scalable multinomial logistic normal estimator for microbial co-occurrence inference. This was made possible by recent advances with VAEs that allowed us to exploit the connection between the multinomial PPCA and multinomial VAEs. This methodology was competitive with other beta diversity metrics, and the performance boost was apparent in longitudinal datasets. Due to the architecture of these models, we can extract co-occurrence information that was obtained from tens of thousands of microbes. And that concludes my talk. Thank you for listening. And I also want to note that next year, I am starting a new group at NIH, and I am hiring. So if you are interested in this type of work, please reach out, and my contact information can be found below. Thank you.